Hello. This project develops and studies a multi-agent model to better understand chemotaxis in heterogeneous environments. I am Daniele Proverbio, and I'll guide you through this poster. Chemotaxis, to begin with, is a gathering strategy by which microorganisms and colonies of them aggregate by following chemical gradients. In particular, in vivo, those microorganism colonies usually live in heterogeneous environments such as forests or water ponds. However, they are still fairly poorly understood because so far models, especially analytical models, have taken, for the vast majority, homogeneous environments. Therefore, there is a need to study what could be a likely development of an aggregation process in heterogeneous environments and develop suitable models in order to study them and test several hypotheses. What we did was indeed to extend a pre-existing multi-agent model of Dictyostelium discoideum, so social amoeba colonies. These models have been published fairly recently and mimics all the gathering mechanisms that are known of the biological system. In this case, each single agent represents a cell with an inner architecture of several behaviors that reproduce what is known of the biology of the cell. The chemical signals are also defined as agents. Everything is embedded into an environment also characterized by the presence of obstacles of different nature. They can be either chemical, therefore just blocking the chemical signal, or they can be physical, therefore blocking the chemical signal and preventing cells from passing through. In order to quantify the results of our in silico experiments, we also defined a measure suitable to tackle evolutions in heterogeneous environments, which is a mean gathering factor. So defining basically the size of the various clusters of cells after the aggregation process. The questions that we ask are, first, whether such decentralized gathering following chemotaxis is possible and robust in heterogeneous environments. Then understanding which are the different types of obstacles or rules that can elicit those kind of behaviors. And finally, to find a minimum set of agents rules that allow the system to maximize this neighborhood score. All in all, to test whether and how robustly simple individual rules might yield complex social behaviors. The results are as follows. Directly from the snapshots of the simulations, you can immediately see that the obstacles act as local sources of perturbation, of topological perturbation in particular, and yield multiple clusters in several subspaces defined by the obstacles themselves. Interestingly, the nature of obstacles doesn't significantly change the gathering process. As you can see, both chemical and physical obstacles yield to very similar patterns in time. As well, we tested individual mechanisms for agents. So either using the simulations as they are, or equipping the agents with further mechanisms to explicitly sense and avoid obstacles. Strikingly, both strategies basically elicit similar gathering behaviors. Finally, we checked for the role of noise, so setting the colonies in such a way that individual fluctuations were very important and relevant in the context of the whole colony. And here we observed that the noise allows for a better space exploration and reduces the likelihood of getting multiple clusters, therefore eliciting the formation of single clusters. All in all, made us understand that, well, first, those kind of models are actually very useful and powerful to reproduce realistic settings, which cannot be tackled by analytical models because of the various challenges. And then, as for the system is concerned, that the streaming patterns are primarily sensitive to the chemical fluxes, not to the nature of the obstacles nor of individual rules, but instead the chemical gradients have an impressive role in directing the gathering process, linking individual rules with social behaviors. In particular, we speculate that specialized sensing mechanisms for obstacle avoidance might be absolutely unnecessary, which could explain why for many organisms, including Dictyostelium, they have not been observed in vivo. And finally, we assessed the noise role in promoting unique clusters, of course, at the price of greater uncertainties during the gathering process. Therefore, speculating about those evolutionary trade-offs. Overall, this study therefore sheds light on several mechanisms and helps generating new hypotheses that hopefully will be eventually tested experimentally. Thank you for your attention. I'll be very happy to get in contact with you with my email or during the conference, and I wish you a nice event.